certainly, cognitive deficits are widespread in neuropsychiatric disorders, as are network disruptions.
Yeah.
And there's a strong, association between what brain regions turn on and when for normal cognition, and those disruptions correlate with the degree of deficits in these different neuropsychiatric disorders. So figuring out what the circuit mechanisms are of network disruption in these different disorders is really important for us to be able to tackle cognitive deficits in these different disorders that are very poorly treated with current therapies.
Yeah. So you see this effect of psilocybin on the circuit linking the anterior cingulate to the claustrum. It depends on 5-HT1B serotonin receptors, not the 2A receptors. And then you get this acute and chronic effect. So the effects outlast the presence of the drug acutely. What are sort of the next experiments here? Where do you see this going from here in terms of disentangling, what's going on here mechanistically for these circuits?
I think, one of the most important things to understand is how the serotonin system is built to modulate the claustrum itself. That will give us insight into understanding how psilocybin is changing the system for potential therapeutic benefit. That's one thing. That's a very complicated thing to tackle because serotonergic innervation of any brain region is super complicated. It's not a simple one-to-one pre to post-synaptic, anatomical microcircuit construction. So that's going to take a lot of effort. Second thing we're really interested in tackling is under what conditions can psilocybin provide therapeutic benefit and under different pathological states. So we can, in what receptors are responsible for that effect. So the interesting thing about this 5-HT1B receptor finding that we had is that adds more evidence to this idea that it's not just activation of the serotonin 2A receptor that might lead to therapeutic benefits. There might be other receptors involved, and they might be involved in different aspects of therapeutic benefit, right? So that's something that we're interested in taking forward.
Yeah. And so what are some of the next things you guys are investigating with respect to claustrum function, whether or not they have to do with the experiments you're doing in alcohol and psychedelics? What are you doing to try and just disentangle the sort of natural endogenous function of this brain region?
Well, the thing we're concentrating on most right now is the effects of chronic alcohol on the claustrum, but we just started another study trying to understand some of the basics of the claustrum, and that goes back to this, the clear difference that people see in the expression of these different network states in the brain. So what we had been talking about is the default mode network turns on when you're in a scanner, thinking about autobiographical things. When you perform a task, a different network will turn on, an extrinsic mode network, let's call it. And these two networks are strongly anti-correlated in the brain. So one will turn on and the other one will turn off, and vice versa. And, when that anti-correlation muddies, you get cognitive deficits. So we think that the claustrum might be built to allow for this anti-correlation effect. So going back to the sort of earlier part of our conversation, let's conceptualize the claustrum as receiving a routing signal from some prefrontal cortex region saying, let's turn on network A.